And we are moving now to Ben Colville, who is a postdoctoral fellow at the Massachusetts General Hospital, so it's Harvard, in Boston, in the United States. And if I understand correctly, Ben is in the group of Professor Jack Szostak. Is that right?
Yeah, that's correct. Can you guys hear me okay?
Yes, we do hear you very well. And Ben will present to us today his work on the synthesis and photochemical purification of threose nucleic acid monomers, so alternative monomers to RNA building blocks. So take it away, Ben.
Great, thank you. Give me 1 second, and hopefully you can see my slides.
Yes, we do see your slides.
Great. Okay, so I'm on the total other spectrum of Albert. I'm very early in the morning here, so bear with me. Okay, yeah, great. Thanks for the introduction, Rafael. So yeah, today I'd just like to talk to you about some of the work that I did at UCL as part of my PhD with Matt Pauner. It's along similar lines to what Albert's been talking about, and it's actually a fantastic introduction for me, so thank you for saving me a lot of talking. And we're also interested in kind of how we can build up complexity from simple prebiotic feedstock molecules into nucleic acids, and then they can be incorporated later downstream into informational polymers. So let's see if we can change slides. Perfect. Okay, so one of the things I think is important is to kind of think about where prebiotic chemistry came from. So that's where we're approaching this problem of complexity from as prebiotic chemists. It's probably kind of Waller's urea synthesis was probably the first prebiotic chemistry reaction, his forming urea. And then not much after that until the work of now famous experiment of Miller and Urey, where they showed that you could take this these gases and subject them to electric spark discharge and form amino acids. I'm sure this is all very well known. And these kind of basic feedstock molecules have then gone on to be shown to incorporate it into proteins and nucleotides, etc. And so as prebiotic chemists, that kind of led us towards the RNA world theory, summed up so well by Albert just a moment ago. And that was kind of really established with the discovery of these ribozymes, so strands of RNA that can catalyze reactions just like the core of the ribosome. And this kind of gave people the idea that RNA was the first genetic polymer to arise because it could function as both the genotype and the phenotype. And so what we would have been interested in is asking if chemistry can tell us if RNA was the first genetic polymer to arise and why. One of the earlier people to work on this problem has been Albert Eschenmoser, and he looked at the kind of the sugar backbone heterogeneity and what that would do to the ability of these genetic polymers to base pair, a fundamental property that allows them to retain and transfer genetic information between them. And so he kind of looked at extensively looked at all of these different sugar backbones, some that paired incredibly well with themselves, like homodNA, but didn't pair with RNA. And that kind of led them to this, this 3,4-lyxo system in the top right. Hopefully you can see my mouse. And this had a shorter than normal backbone length. So RNA and DNA have a 6-atom backbone, but this 3,4-lyxo compound only had 5 atoms. And that kind of led them to this other simpler system, the threose nucleic acid system, which also has a 5-atom backbone repeat. What was so fascinating about TNA was that not only could it base pair well with itself, it could, Watson Crick Franklin base pair very efficiently with RNA and DNA. And due to these properties and the fact that it's constitutionally simpler, so it can be comprised of only 2 C2 sugars rather than a C2 and a C3 sugar like RNA must be, people have been wondering whether this could have been a potential progenitor to RNA for a number of years. Incidentally, just some other interesting things about threose nucleic acid as a system. It can only form 2', 3' linkages, whereas RNA, we know well that it forms these 5', 3' linkages, but it can also form these 5', 2' linkages, which can be a problem. Additionally, TNA, due to its lack of 2' hydroxyl, is very hydrolytically stable, comparable to that of actually DNA, much more stable than RNA. And this is presumably due to the fact that RNA can undergo this intrastrand cleavage, forming these 2,3 cyclic phosphates, which will become important later on. And so we were interested in approaching this problem of is RNA the first genetic polymer? Could TNA have come before? And fundamentally, we approached it from a prebiotic chemistry perspective. So can we make TNA? If we can't make it, then it's probably not going to be relevant. And as we've heard this morning or this afternoon already, these kind of dry down reactions, taking the sugar and the base and drying them down to make nucleosides, just doesn't work for a number of reasons explained here. So, I mean, one of the other problems is that we don't get much ribose in these kind of formose systems, and this ribose actually prefers to sit in its pyranosyle form. And then you have the problem of pyranosyle glycosylation. If you do have these reactions working, you get a mixture of these sugars as well. And so an answer to this was provided in kind of 2009, where you could take these simple molecules that Albert's shown you that you can produce really well, glycoaldehyde, cyanamide, and 2-aminoxazole, the first two here, and make arabinosaminoxazoline, cyanovinylate, phosphorylate to give you cytidine, activated cytidine. And what was interesting as well is that they showed that this can be photochemically hydrolyzed from cytidine to uridine, so you get 2 canonical nucleotides for the price of 1. And so we were really interested to see whether TNA can be synthesized under conditions that also yield RNA. One of the interesting things about this synthesis of RNA was the chemistry that they used. It gives you a variety of diastereomers at the aminoxazoline stage. So not only do you get the arabinoaminoxazoline that you need to make the activated cytidines that were shown in 2009, you also get the ribo, the xylo, the lyxo, and the pyranosyle lyxo. Additionally, you have unselective reactivity to deal with. So you have the combination of cyanamide and glyceraldehyde, that gives you this dead end product here. And also the equilibrium of glycoaldehyde sits way in favor of dihydroxyacetone, its more stable ketose compound. And so we thought maybe en route to TNA, we could hopefully use a similar aminoxazoline approach, but maybe it would actually solve some of these problems too. And so some earlier work had shown that you can very efficiently make these the 2 diastereomers of the tetrose series, so the threose that we want, and the erythrose, which also comes along for the ride. And so you can make this in greater than 90% yield very efficiently. And one thing that we were interested in is, can we do this in one pot? Because conceptually, you can just take 2 molecules of glycoaldehyde, one to make the 2-aminoxazole, and then one to make the tetrosaminoxazolines. And gratifyingly, we showed that when you kind of do this at kind of high pH, you can get a decent amount of these tetrosaminoxazolines. Additionally, the selectivity of these reactions is quite fascinating. So this is from some earlier work from our lab. So when you take a mixture of equilibrated glyceraldehyde and glycoaldehyde and expose it to 2-aminoxazole, you selectively get way more of the tetrosaminoxazolines than you do of the pentosaminoxazolines or the apoaminoxazolines. So kind of all routes seem to lead us towards TNA. So we were very optimistic that if we could use aminoxazolines, we were on the right route to make TNA. So this is kind of where we went. And so, as we said earlier, we have these arabinoaminoxazolines that can make ribonucleotides, but in recent years, we've seen that the use of sulfur and photochemistry has allowed the Sutherland group to transfer ribose aminoxazoline into the desired, to also incorporate ribose into these prebiotic networks. And so we were hopeful that we could use something similar. So we started off looking at the cyanovinylation, and this is very efficient. So we were easily able to convert the tetrosaminoxazolines into their respective tetrose and hydronucleosides. So we're working with both of the diastereomers here, and that will be important later on. So with these anhydronucleosides in hand, we next looked at their thiolysis, because as we've seen, positioning sulfur selectively at the C2 position of these nucleobases can confer a great increase in their photochemistry. So this thiolysis proceeds in upwards of 75% yield overnight, just at room temperature, to give us both of these beta orientated tetroses, the threo and the erythro. And so we have the right compound, we just have it in the wrong conformation. An interesting aside here is that we can actually use hydrogen peroxide to rapidly recycle or recyclize these compounds and prevent them from undergoing this slow hydrolysis to these the oxygenic cytidines, which will become important momentarily. And so next, we wanted to look at their photochemical behavior, and gratifyingly, these things photoanimerize very efficiently. They kind of reach a photostationary point after about 2 or 3 days, and they also selectively kind of destroy the starting material, so you kind of only end up with your desired product. So you can see the green and the orange are left after the 3 days here, and then this is just subsequent spiking experiments to confirm we've made what we think we've made. So that was excellent. So we kind of got the right sugar and the right conformation, and we've shown that hydrogen peroxide can oxidize these to the oxygenic versions, the natural versions. And so this is the kind of current prebiotic, the current picture of the prebiotic tetrose reactivity. So we can thiolyze these anhydronucleosides, we can recyclize them to protect them, which is important because the oxygenic cytidines don't undergo efficient photochemistry. They instead form these oxazolidinones. But you know, if we do the UV photochemistry, flip them, and then we can remove the sulfur. And this was great, but we'd only managed to make the threo and the erythro together. And we thought maybe there was a way that we could do some sort of selection to selectively get the threose one and purify it away from the erythros one. And so we turn to phosphorylation, which is kind of an essential activating component. 2 or 3 minutes?
Yep, about 3 minutes.
Thanks. So yeah, we turn to phosphorylation to see if this could provide an answer for us. What was really interesting is because of the cis-vicinal diols of the erythrodiosteromer, it really selectively and efficiently forms these 2,3-cyclic phosphates. And then in the beta form, once irradiated, we saw total destruction after a very short time period, 1 or 2 days of radiation. And the only thing we observed was the base, so 2-thiocytosine. So something was happening that was forming this base to fall off. And it's probably something to do, something like this. So we very selectively form the 2,3-cyclic phosphate, and then once we irradiate, the base efficiently flips like it did when it was just a nucleoside. And then this kind of sets up a situation where you have like a it's kind of primed for an intramolecular SN2 reaction and can attack itself, and then facilely hydrolyzed to give you the base and the sugar phosphate. And so this was really exciting because this kind of gave us a handle to be able to purify this reaction. So when you take threo and the 2,3-cyclic phosphate here, for example, you can selectively get just the threo after a number of days and just the nuclear base left. So you can totally destroy all of the erythrodiosteromer. And this also works when you have phosphorylated threo. So this kind of led us to a situation where we could have this kind of sequence agnostic construction. So you can have threo and erythro together, and you can phosphorylate them or radiate them in kind of any order, because all routes lead to phosphorylated pure threose nucleotides. And so just to sum up, so conceptually TNA is kind of structurally simpler than RNA, but was it generationally simpler? We think it's just as generationally plausible as RNA if we're invoking a photochemical synthesis of RNA. And so for that reason, I think any scenario which invokes this photochemical synthesis of RNA must also consider the possibility of TNA and threose nucleic acid being present. And with that, I'd just like to thank Matt Powner at UCL, my supervisor, number of other members of the Powner Group, the Simons Foundation for the funding, and I'd like to thank Jack Shostak and my current funding bodies for their assistance for that. And thank you all for listening.
Excellent. Thank you, Ben. Really, really interesting. So with this reaction sequence, you can get 2 out of 4 TNA nucleosides. Is that correct? So
certainly you can get the cytidine one. I think we're hopefully going to be able to show that you can get uridines pretty simply from that too. But that's still ongoing work. Yeah.
So I guess an interesting question is it possible, or I mean, have you been thinking about taking the synthesis further and trying to do something analogous to the recent works from the Sutherland group, which take into account thioanhydronucleosides? So you use anhydronucleoside precursors, and if we consider thioanhydro, maybe we could actually get to the purines at some point.
You mean doing the transglycosylation of the anhydros with thiopurines? Yes. When I saw that, I was like, that seems like it would be very applicable here, and is super exciting. Hopefully, not something I'm currently working on, but I do have hope that somebody in the Pounder group will take it up, or maybe I'll get a chance to work on it later down the line.
Very interesting. We have a question from Albert. So Albert is writing, thanks for the amazing talk, Ben. Have you ever tried taking the D-threoanhydrocytidine isomer and dry that down with phosphate to make the cyclic TNA phosphate without H2S?
So I don't quite understand. Do you mean when the vicinal diols? So the erythrodiosteromer, is that where he's getting at?
Good question.
Maybe he can answer. But we did try that, and you do just, it doesn't really work that well. I think the sugar pucker doesn't seem to quite be the same as for the ribose system, which obviously you can put it on the 3 and it'll transfer and kick out the base. It doesn't seem to quite work so well with these tetrose sugars. I think that's what he means.
Okay, thanks, Ben. So I was also wondering how, or maybe let me phrase it differently. Are there many works on the structure of TNA, just to think about how a polymer like this could behave, and could it actually possibly, I mean, by looking at the structure, could it be possible for it to play any biological or biochemical functions similar to what RNA does?
I think there's a really exciting possibility of people using it in like antisense oligonucleotides. So it can, because it binds really well with RNA. But in terms of its genetics, I think people have looked at how it can be used in non-enzymatic primer extension, and the structural implications of incorporating through nucleotides. Is that what you're getting at?
Yeah, I mean, I'm just curious. Imagining a TNA world, could because of some reasons in alternative biology, TNA play the role of RNA, for example?
Well, I think the lack of the 2' hydroxyl kind of leads you into a similar place as DNA, where you're resorting to a different form of like threozyme catalysis, where you're much more reliant on kind of maybe heterogeneous bases with different functionalizations, and you're not so reliant on having that 2' hydroxyl to do stuff like that. But it is certainly interesting. I think for us, the interesting thing is thinking about these kind of chimeric genetic polymers like Ramkrishna Murthy has looked at, and how those kind of behave, rather than just a pure TNA system.
Thanks. So Albert just followed up. So he's writing like just dry down with phosphate, like the original Orgel demonstration, except with the arabinose aminoxazoline.
The arabinose aminoxazoline. So I haven't personally done those reactions with the pentose aminoxazolines. I haven't looked at I can't remember what happens when I don't think I'd ever looked at the tetrose aminoxazolines under those kind of dry down phosphorylation conditions too. But certainly something to consider.
Okay, thank you so much. Thanks, Ben. Really nice to have you here. And
Thanks very much. Good to see you, Rafael.
Thank you. Good to see you too.